As part of this topic called titled Transport in Animals. So, in this part, we'll be looking at the blood, component of the blood. So, the learning objective for this lesson include for us to be able to list components of the blood, which include the red blood cells. We'll look at white blood cells, platelets, and plasma. Then we should be able to identify white blood cells as seen under a light microscope. Also state the functions of the part or the different component of the blood. Then we'll be able to identify lymphocytes and phagocytes. Then we should be able to state the functions of both lymphocyte and phagocyte. Lymphocyte in antibody production and phagocyte in phagocytosis. Then we should be able to describe the process and importance or role of blood clotting, and also to describe the materials, the transfer of materials between capillaries and tissue fluids. So, blood component. So, the blood itself consists of red blood cells, white blood cells, platelets, and plasma. So, if you look at the component of the blood and you separate each component, you find out that 55% of the blood is made up of plasma, and the plasma, this is the liquid component of the blood that helps in transport of blood cells, ions, soluble nutrients, hormones, and carbon dioxide. That's their major function. They helps in the transport of all the other components, including the blood cells, red and white blood cells, ions, soluble nutrients, hormones, and carbon dioxide. Now, the other 45% is what makes up the red blood cells, white blood cells, and platelets, makes up 45% of the component or composition of the blood. And the white blood cells, major function, it helps in defense through the process of phagocytosis, through phagocyte and antibody production through the lymphocyte. While we also have platelets, and they are responsible for blood clotting. The red blood cells help in the transporting of oxygen around the body using hemoglobin, which is the pigment that bind to oxygen to form oxyhemoglobin that helps in the transport of oxygen around the body. Now, looking at a photomicrograph, and in most IGCSE questions, I've seen questions where they'll give you a picture just like the one that is here, and they expect you to be able to identify the red blood cells. And you see the red blood cells do not have nucleus, and they are disc, biconcave disc shape, and they don't have nucleus, so they will be able to transport high amount of oxygen. So we have red blood cells, and these are white blood cells, and in most cases these are phagocytes because they have a lump nucleus. Now, in the case where you eventually see a lymphocyte, lymphocytes have a large nucleus at the middle. So in the case of lymphocytes, you see something like this. Let me try and draw it. In lymphocytes, you see circular. However, there is a large nucleus at the middle in the case of lymphocytes, which is also a type of white blood cells, which we'll look at soon. We have platelets, and the liquid component outside is the plasma. So quickly, let's look at white blood cells. Now, there are two types of white blood cells. White blood cells are part of the body's immune system, and its major function is for defending against infections by pathogenic microorganisms. So pathogens are microorganisms that causes diseases. And there are two types of white blood cells. We have majorly the phagocytes, and this is a phagocyte. Now remember, I said it has a lump nucleus at the middle. A lump nucleus. why the next one here is a lymphocyte, and it has a large nucleus at the middle. So that's how you should be able to differentiate the two type of white blood cells. Okay, now phagocytes, how do they carry out their function of phagocytosis? You need to be able to explain this process because it comes out a lot also in exams. And you see, it carry out phagocytosis by engulfing and digesting pathogens. So you should take note of this major keyword. You see, by engulfing and digesting pathogens. That is how we carry out the process of phagocytosis. So first, if you have a bacteria pathogen, now this is a pathogen and this is a phagocyte. Remember, it has a lone nucleus and these are mitochondria and mini vesicles here and endoplasmic reticulum. Now, what happens is the phagocyte will identify the pathogen. So, bacteria being engulfed. So, once you identify the pathogen, it will now move towards it and engulf it. Engulf it. So, bacteria being engulfed by phagocytes. That is phagocytosis. Now, the inside the phagocyte, you have digestive granules, which produces digestive enzymes. Even the lone nucleus produce digestive enzymes. So the bacteria will now be enclosed in a phagosome. Now, this is a phagosome, and the bacteria is enclosed in it. So the granules of digestive enzymes will now digest or break down this bacteria to now destroy it, that it will no longer be harmful. And that's just the process involved in phagocytosis. Now, phagocytes have a sensitive cell membrane that can detect chemical produced by pathogenic cells. Once they encounter the pathogenic cells, they will engulf it and release digestive enzymes, to digest it. They can be easily recognized under the microscope by multi-lobed nucleus. They can be recognized under the microscope by their multi-lobed nucleus and their granular cytoplasm. So, if you're asked, though, in the exam, describe the process of phagocytosis, simple. Phagocyte will recognize a pathogen and move towards it. Once it moves towards it, it will engulf it into it. It will engulf it, and the bacteria will be enclosed inside a phagosome, and it will release digestive enzymes which will be used to destroy the bacterial cells. Now, this is more like what happens. It recognizes it and moves towards it and engulf it inside its phagosome. So this is now the phagosome. So digestive enzymes will now help to break down the pathogens inside to now make it harmful. It engulfs it. Look at digestive enzymes. We now act on it to help break it down, into a harmless pathogen. Okay, lymphocytes. Uh, lymphocytes is the second type of white blood cells that aid in the process of producing. So it produces antibodies. So take note, there is a difference between antibody and antibiotics. Antibody is not the same thing as antibiotics. Antibiotics are only used to kill, are only used to destroy bacteria. So antibiotics simply means antibacterial. So it's different from antibodies because antibodies aid to destroy pathogenic cells, and it also produces antitoxins to neutralize toxins released by pathogens. So it has two functions. Lymphocyte produces antibodies to destroy pathogenic cells and antitoxins to neutralize toxins released by pathogens. Now, they can easily be recognized under the microscope by their large round nucleus, which take up nearly the whole cell, and their clear non-granular cytoplasm. So remember we looked at it here. They have a large clear nucleus, and they produce antibodies and antitoxins. So when we look at disease and immunity, we'll now explain how the antibodies, which produces a Y-shaped cells, help in agglutination of most pathogens. So we explain that in much more detail when we look at disease and immunity, but this is the aspect you need to know for this topic. Now, next we'll be looking at blood clotting. Blood clotting. Now, this is a process that is carried out majorly by platelets. Okay, it's here. By platelets. So platelets are fragments of cells which are involved in blood clotting and forming scabs where skin have been cut or punctured. Now, blood clotting prevent. Now, when you ask the role of blood clotting, this is it here. Role of blood clotting is this. So this is the role of blood clotting. Role of blood clotting. Take note. If you ask what is the role, because you need to know both the role and also how the process. So blood clotting help to prevent loss of blood, continued loss of blood from the wound. Now, the scab that is formed help to seal the wound with an insoluble patch, and that help to prevent the entry of microorganisms. So it helps to prevent continuous loss of blood, and it also helps to prevent entry of microorganisms. Now, it remains in place until a new skin has grown underneath, so sealing and the skin again. So process of blood clotting. First, when the skin is broken, that means there is a wound, platelets arrive to stop the bleeding. Now, a series of reactions occur within the blood plasma as platelets arrive. So the first thing is platelet release chemical that causes soluble fibrinogen. Take note, this is the major stuff you need to know. Platelet release chemicals that causes soluble fibrinogen protein to be converted to insoluble fibrin. Now, this insoluble fibrin and form an insoluble mesh. Insoluble fibrin will now form an insoluble mesh around the wound by trapping red blood cells and therefore forming a clot. Simple. So, platelet release chemical that convert soluble fibrinogen to insoluble fibrin, which form an insoluble mesh on the cut wound by trapping red blood cells. Now, the clot eventually dries and develops a scab to protect the wound from bacterial entry. So, here, once you have a broken wound first, and the platelets, we have soluble fibrinogen proteins here. So, this is soluble fibrinogen proteins. The platelet release chemical to convert them into insoluble fibrin. So you see the platelet releasing chemicals. So it will convert it to insoluble fibrin, which will now trap red blood cells to form a mesh. So it is now activated to form a mesh along the cut surface to stop the loss of blood. So thank you. I think we're done with this part. So